Hello everyone, welcome to my channel. This is Dr. K. Srinivas Reddy, Assistant Professor of Botany, Government Degree College for Women Nalgonda. In my previous video, I have explained about nucleic acids, types of nucleic acids, and the chemical constituents of nucleic acids, nucleosides, and nucleotides. In this video, I am going to explain about the structure of DNA. Okay. In this video, I am going to cover structure of DNA, types of DNA, and functions of DNA. Okay, let us discuss about the structure of DNA. DNA stands for deoxyribonucleic acid. Deoxyribonucleic acid. DNA is a polymer, or you can say macromolecule. It stores the genetic information and play an important role in heredity. As you all know that transformation of characters or traits from parental generation to offsprings is known as heredity. In heredity, DNA plays very important role as it stores the genetic information. Okay. Watson and Crick proposed the double helix model of DNA in 1953. By using the data of experiments of various scientists such as Wilkinson, Franklin, and Chargaff. Based on the data of the previous experiments, Watson and Crick proposed the double helix model of DNA in the year 1953, and the model was widely accepted. And coming to the experiments which were conducted by Wilkins and Franklin, MHF Wilkins and Franklin Rosalind conducted X-ray crystallography of DNA in the year 1950. In X-ray crystallography, X-rays are fired at DNA sample and the scattered rays from the DNA sample is observed by catching them on the photographic film where X-rays produce spots. On photographic film, rays scattered from the DNA sample produce spots. By observing these spots, Wilkinson and Franklin constructed the three-dimensional structure of DNA. Three-dimensional structure of DNA. In X-ray crystallography, X-rays are fired at DNA sample and the rays scattered from the DNA sample is observed by catching them on the photographic film where X-rays produces spots. By conducting X-ray crystallography of DNA, Franklin and Wilkins proposed some points. Here we are going to discuss about the points which were proposed by Wilkins and Franklin. Here the first point is DNA molecule is a multi-stranded fiber. DNA molecule, here you can see, DNA molecule is a multi-stranded fiber. It means the DNA is made up of more fibers. It consists of more fibers. Okay. And the diameter of the molecule was about 22 angstroms. 22 angstroms. The diameter of the DNA molecule about 22 angstroms. And the DNA molecule have 34 angstroms gaps along the length of the fiber. Along the length of the fiber, the gaps are present, and the gaps are measured about each gap measures about 34 angstroms in length. 34 angstroms in length. Okay. And the DNA molecule has repeating units. DNA molecule has repeating unit. This is the fourth point which was proposed by Wilkins and Franklin. And the point is that DNA has the repeating unit, and the space between or the gap between the two successive repeating units is about 3.4 angstroms. 3.4 angstroms. So based on these findings, Wilkins and Franklin proposed or constructed a three-dimensional structure of DNA. The points which were proposed by Wilkins and Franklin are: the DNA molecule is multi-stranded fiber, and the diameter of the DNA molecule was about 22 angstroms, and the DNA molecule have 34 angstroms gaps along the fiber, and the DNA molecule has repeating units, and there is a 3.4 angstrom space in between two successive repeating units. Okay. Before these experiments, Erwin Chargaff in 1940 proposed a rule that is called Erwin Chargaff's rule, or it is also known as Chargaff's rule of molar equivalence. Chemical analysis of DNA molecule by Chargaff is revealed that, here you can see the points. The first point is that the number of purine bases is equal to the number of pyrimidine bases. The sum of adenine and guanine is equal to the sum of cytosine and thymine. The number of purine bases in a DNA molecule is equal to the number of pyrimidine bases. Okay. And second point, the quantity of adenine is equal to thymine. The quantity of adenine is equal to thymine, and the quantity of guanine is equal to cytosine. The quantity of adenine is equal to thymine. The quantity of guanine is equal to cytosine. Okay. And the third point is adenine is always paired with thymine with two hydrogen bonds, and vice versa. Adenine is always paired with thymine with two hydrogen bonds, and vice versa. Similarly, guanine is always paired with cytosine with three hydrogen bonds, and vice versa. And this type of pairing is known as complementary base pairing. Complementary base pairing. Adenine always pairs with thymine with two hydrogen bonds, and vice versa. Similarly, guanine always pairs with cytosine with three hydrogen bonds, and vice versa. So this type of pairing is known as complementary base pairing. And these points are very, very important to understand the structure of DNA. The points which was proposed by Erwin Chargaff by chemical analysis of DNA, and also the points which were proposed by Wilkins and Franklin by X-ray crystallography of DNA. With the help of these findings, Watson and Crick proposed the double helix model of DNA in the year 1953, and it is very important discovery like Origin of Species by Charles Darwin. For their outstanding work, Watson, Crick, and Wilkins shared Nobel Prize in 1962. Watson, Crick, and Wilkins shared Nobel Prize in the year 1962. So it was one of the important breakthrough in biology. Okay. Next, coming to the structure of DNA, which was proposed by Watson and Crick. Okay. Let us discuss about the main features of Watson and Crick proposed model of DNA. Here, the first point is DNA is made up of two polynucleotide chains. Here you can see in the picture two polynucleotides. This is one polynucleotide chain and this one is the other polynucleotide chain. Okay, these are the two polynucleotide chains present in DNA molecule. And each polynucleotide is composed of many nucleotides. Each polynucleotide is made up of many nucleotides, hence it is called polynucleotide. Poly means many. Many nucleotides are present in each chain, that is polynucleotide chain. Here you can see different types of nucleotides present in each polynucleotide chain. Here you can see this is one nucleotide. This is another nucleotide. This is one more nucleotide, and this one more nucleotide, and so on. In this way, many number of nucleotides are present in the chain, in each chain. Here also, number of nucleotides are present in this chain also. So these two chains are called polynucleotides or polynucleotide chains. Polynucleotide chains. Each chain contains many number of nucleotides. Hence, these chains are called polynucleotides. Two polynucleotides are present in each DNA molecule. And the third point is that the two strands are oriented anti-parallel to each other. Here you can see one strand runs in 5 to 3 prime direction and the other strand runs in 3 to 5 prime direction. 5 to 3, 3 to 5 direction. So this type of arrangement is called anti-parallel orientation. Two strands are arranged anti-parallel to each other. Anti-parallel to each other. One strand runs 5 to 3 prime direction and the other strand runs in 3 prime to 5 prime direction. 5 prime direction. And the fourth point is that anti-parallel orientation is essential for the formation of hydrogen bonds in between the nitrogen bases of two opposite polynucleotide strands. If anti-parallel orientation is not there, it is not possible to form hydrogen bonds in between the nitrogen bases of two opposite strands. Okay. Here you can see these are the hydrogen bonds. Adenine pairs with thymine, thymine pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine with hydrogen bonds. So this is possible due to reverse orientation, anti-parallel orientation of two polynucleotide strands only. And the fourth point is that adenine is linked by two hydrogen bonds with thymine. Adenine is always paired with thymine with two hydrogen bonds. Here you can see in the picture, this is adenine and this one is the thymine, and these are the two hydrogen bonds in between adenine and thymine. This adenine belongs to this polynucleotide chain and this thymine belongs to this polynucleotide chain. Okay, adenine of one strand paired with thymine of the opposite strand with two hydrogen bonds, and vice versa. In the same way, guanine is pairs with cytosine by three hydrogen bonds and vice versa. Guanine of one polynucleotide strands pairs with the cytosine of opposite strand with three hydrogen bonds, or vice versa. Here you can see vice versa. Here cytosine present on this polynucleotide chain, guanine present on the opposite polynucleotide strand, and three hydrogen bonds are present in between these two nitrogen bases of two opposite strands. In this way, base pairing is taking place in between the nitrogen bases of two opposite strands, two polynucleotide strands which are oriented anti-parallel to each other. So these are the very important point to understand the structure of DNA. DNA is made up of two polynucleotide chains, okay? And each polynucleotide is composed of many nucleotides. More number of nucleotides are present in each polynucleotide chain, hence the name is polynucleotide. Many number of nucleotides present. And the two strands are arranged anti-parallel to each other, oriented anti-parallel to each other. One strand runs 5 to 3 prime direction and the other strand runs 3 to 5 prime direction. And this anti-parallel orientation is very essential for the formation of hydrogen bonds in between the nitrogen bases of two opposite strands of DNA molecule. Anti-parallel orientation is very important for the formation of hydrogen bonds between the nitrogen bases of two strands of a DNA molecule. Next point is that adenine always pairs with thymine. Adenine of one polynucleotide strand is pairs with the thymine of opposite strand with two hydrogen bonds and vice versa. It means thymine of one polynucleotide strand pairs with adenine of the opposite strand with two hydrogen bonds. And similarly, the guanine of one polynucleotide strand or chain is paired with cytosine of the opposite strand with three hydrogen bonds and vice versa. And this type of pairing is called complementary base pairing. Complementary base pairing. Okay. And next point is that the two strands are coiled together in a right-handed helix, forming the double helix of DNA. Here you can see in previous slide you see the two anti-parallelly arranged polynucleotide strands. Here in this picture the two strands are coiled together in right-handed helix, manner. In right-handed helix. In this picture here you can see this is right-handed coiling of DNA molecule. The two strands of DNA coiled along their longitudinal axis in a right-handed direction or in a right-handed manner and forming the DNA double helix. And next point is that the diameter of the helix is about 20 angstroms and the length of each coil is 34 angstroms. Here you can see the diameter from here to here. From here to here. The diameter from here. This is the diameter. The diameter of the helix is about 20 angstroms. And the length of each coil is about 34 angstroms. Here you can see from here to here, from here to here. This is a coil, complete coil, which rotates 360 degrees, starting from here to here, 360 degrees, and the length of this turn or coil is about 34 angstroms. 34 angstroms. and each coil contains 10 equally spaced nitrogen base pairs. Here you can see from here to this is a nitrogen base pair step, and this is another base pair step, one more base pair, one more base pair steps. So base pairs are arranged like steps in the ladder in between the two strands of polynucleotides of DNA molecule. Here you can see these are the steps. Nitrogen base 10 equally spaced nitrogen base pairs are present in each coil. Equally spaced, and the space between the two successive base pairs is 3.4 angstroms. Here you can see in the picture the space between from here to here, 3.4 angstroms. 3.4 angstroms. The space between, or the gap between, two successive nitrogen base pair steps is 3.4 angstroms. Total 10 nitrogen base pairs are present in a coil, in a helix, or you can say in one helix. 10 nitrogen base pairs are present and they are equally spaced, and the space between, or the gap between, two successive nitrogen base pairs is 3.4 angstroms. 3.4 angstroms. Here, each turn or coil include one major groove and one minor groove. This is the major groove from here to here. This is the major groove and this is the minor groove. This one is the minor groove. This is the major groove. Major groove and minor groove. Each coil or each turn, each coil or each turn consists of two grooves. One is major groove and another one is minor groove. In this way, DNA is made up of two polynucleotide strands. They are arranged anti-parallel to each other. Polynucleotides are oriented anti-parallel to each other, and these two strands coiled together in right-handed direction along the long axis. This is the axis of the DNA molecule, long axis of the DNA molecule. As a result, a DNA double helix is formed, and the diameter of the helix is about 20 angstroms, and the length of each coil or turn is about 34 angstroms. 34 angstroms in length, and each turn or coil consists of 10 equally spaced nitrogen base pairs. 10 equally spaced nitrogen base pairs. And the gap between the two successive base pairs is about 3.4 angstroms. 3.4 angstroms. And the angle between the two nitrogen base pairs is 36 degrees. 36 degrees. And each turn or coil include one major groove and one minor groove. Okay. Let us discuss about the types of DNA. 4 types of DNAs have been identified so far. They are: number 1, bDNA, number 2, aDNA, number 3, zDNA, number 4, cDNA. 4 types of DNAs have been identified so far: bDNA, aDNA, zDNA, and cDNA. Let us see the comparative features or characteristics of these four DNAs. Here these are the DNAs, different types of DNAs, bDNA, aDNA, zDNA and cDNA, and these are the characters. The characters are coiling, length of each turn or coil, base pairs per coil, diameter of the helix, space between the nitrogen base pairs or two successive base pairs, and sugar phosphate backbone. bDNA, which is the most commonly found DNA, here the coiling is right-handed, and the length of each turn is, or each coil is about 34 angstroms, and base pairs per turn is 10. 10 base pair nitrogen base pairs are present in each coil, and the diameter of the helix is 20 angstroms, and the space between the two successive pairs of nitrogen base pairs is 3.4 angstroms, and sugar phosphate backbone arrangement is regular form. So this is the most common type of DNA, bDNA. The structure which proposed by the Watson and Crick is bDNA. Coming to the second type of DNA, that is aDNA. The coiling is right-handed here also, and the length of the each turn is about 28 angstroms, and the base pairs present in the each coil is 11, and the diameter of the helix is about 23 angstroms, and the space between the two successive base pairs is 2.56 angstroms. The space between the two successive nitrogen base pairs is about 2.56 angstroms. Coming to the zDNA, which is the left-handed DNA. The coiling is left-handed orientation. Coiling is left-handed, so which is entirely opposite to bDNA. Left-handed orientation, and the length of each turn or coil is about 45 angstroms. The length of each coil is 45 angstroms, and the number of base pairs present in each coil is 12. And the diameter of the helix is 18 angstroms, and the space between the two successive nitrogen base pairs is 5.71 angstroms, and sugar phosphate backbone is in zigzag arrangement, found in zigzag. Coming to the cDNA, it is also right-handed form of DNA, and the length of the helix is about 31 angstroms, and the base pairs present, number of base pairs present in each helix is 9.33, and the diameter of the helix is 19 angstroms, and the space between the two successive base pairs is 3.32 angstroms. Sugar phosphate backbone show regular mode of arrangement. Regular mode of arrangement. In examination point of view, they may ask like this: write about the differences between bDNA and zDNA as a short question. Here, if you see the differences between bDNA and zDNA, or it is the right-handed coiling DNA, zDNA is the left-handed coiling DNA, and the length of the helix is 34 angstroms. Length of each helix is 34 angstroms, whereas here the length of each helix is 45 angstroms, 11 angstroms more. And the number of base pairs present in each helix or turn is 10, and here 12. 12 nitrogen base pairs are present for each turn. And the diameter of the each turn is 20 angstroms in bDNA, and the diameter of the helix in zDNA is 18 angstroms. 18 angstroms. and the space between the two successive pairs is 3.4 angstroms. 3.4 angstroms. In case of ZDNA, the space between the two successive pairs is 5.71 angstroms. 5.71 angstroms. Sugar phosphate backbone show regular mode of arrangement in case of BDNA, and in case of ZDNA, sugar phosphate backbone show zigzag arrangement. Zigzag arrangement. These are the main differences between BDNA and ZDNA, which is important for your academic semester and examinations. Okay, let us discuss about the functions of DNA. DNA performs two important functions. They are number one, autocatalysis, number two, heterocatalysis. Let us see what is autocatalysis. The process of duplication of single DNA into two daughter DNA molecules is known as autocatalysis. The process of duplication of single DNA molecule into two daughter DNA molecules is known as autocatalysis or replication. DNA replicates by means of semi-conservatory mechanism. As a result, daughter DNA molecules contains one old strand and one new strand. One old strand is the parent strand and one newly synthesized strand. So that this mechanism is called semi-conservatory mechanism. Coming to the heterocatalysis, DNA promotes the synthesis of proteins and also regulates various biochemical pathways. In this process, the DNA template transfers the message to mRNA by a process called transcription. The DNA template transfers the message to mRNA by a process called transcription. Transcription is the process of formation of mRNA. In other words, we can say from the DNA template. The process of formation of mRNA from the DNA template is known as transcription. And mRNA synthesizes proteins based on the information which is present on it by a process called translation. The DNA template transfers the message to mRNA. Based on the information available on mRNA, proteins are synthesized, and the whole process is called central dogma. In this way, DNA performs two very important functions: autocatalysis and heterocatalysis. Autocatalysis is the process of duplication of single DNA into two daughter DNA molecules. Heterocatalysis, DNA molecule promotes the synthesis of protein and regulates biochemical pathways. In this process, the DNA template transfers the message to mRNA by a process called transcription, and proteins are synthesized based on the information which is available on mRNA by a process called transcription. Both transcription and translation together known as central dogma. Okay, so these are the two very important functions performed by DNA molecule. In this video, I have covered the structure of DNA and the types of DNA and the functions of DNA. Coming to the structure, I have covered the model which was proposed by Watson and Crick. Watson and Crick proposed double helix model of DNA. According to Watson and Crick, each DNA consists of two polynucleotide strands, and these polynucleotide strands contain many number of nucleotides, and these two polynucleotide strands are oriented antiparallel to each other. And antiparallel orientation is very important for the formation of hydrogen bonds between the nitrogen bases of two opposite strands. Okay, and adenine of the one polynucleotide strand pairs with the the adenine always pairs with the thymine of the opposite strand with two hydrogen bonds, and vice versa. It means thymine pairs with the adenine with two hydrogen bonds. And similarly, guanine pairs with cytosine with three hydrogen bonds, and vice versa. Okay. In this way, the two strands arranged oriented antiparallel to each other, and the hydrogen bonds are formed in between the nitrogen bases of two opposite strands, and these two polynucleotide strands coiled together in right-handed manner, coiled together in right-handed manner. As a result, DNA double helix is formed. DNA double helix is formed. The diameter of the helix is about 20 angstroms, and the length of each helix is 34 angstroms. Each helix composed of 10 equally spaced nitrogen base pairs. Nitrogen base pairs, equally spaced nitrogen base pairs. And the space or the gap between the two nitrogen bases is 3.4 angstroms, and the angle between the two successive nitrogen base pairs is 36 angstroms. 36 angstroms. Each turn or each coil include one major group and one minor group. One major group and one minor group is present in each coil or in each turn. Okay. And later on, we discussed about the different types of DNAs. Four types of DNAs so far identified: bDNA, aDNA, zDNA, and cDNA. Except zDNA, remaining three types of DNAs are right-handed coiled DNAs. Right-handed coiled DNAs, and in our remaining three DNAs, the mode of arrangement of sugar phosphate backbone is regular, whereas in zDNA it shows zigzag arrangement. In zDNA, sugar phosphate backbone show zigzag mode of arrangement. After that, I have covered the functions of DNA. DNA perform two important functions. One is autocatalysis and heterocatalysis. One is autocatalysis and second one is heterocatalysis. Autocatalysis is the process of duplication of DNA, single DNA molecule, into two daughter DNA molecules. It is also known as replication. DNA replicates by means of semi-conservatory mechanism. As a result, the daughter DNA molecules possess one old strand and one new strand. Coming to the heterocatalysis, the DNA molecule promotes the synthesis of proteins and controls biochemical pathways. In this process, the template strand of DNA transfer message to mRNA by a process called transcription, and the information which is present on the mRNA initiates the synthesis of proteins. Proteins are synthesized based on the information which is available on mRNA by a process called transcription. Both transcription and translation together known as central dogma, from DNA to mRNA, mRNA to protein. The formation of mRNA from DNA is called transcription, and the synthesis of proteins based on the information present on mRNA is known as translation. Is known as translation. So with this I stop my presentation here. Thank you one and all. Thank you for patiently watching my presentation, and we will meet in my next video. Next video. In my next video, I'm going to explain types types of mRNAs, so types of RNAs and their functions. Right now, bye bye. Thank you one and all.